So today I am very excited that we have Diana Garcia. She's a PhD student in biomedical sciences at the Theoretical Oncology Lab at the National Institute of Genomic Medicine in Mexico. And her main research focus is co-expression cancer networks, and she aims to promote participation of Mexican students, especially women, in network science. So, Diana, take it away.
Thank you, Alison. Thank you for having me. So the title of this talk is Loss of Range Gene Co-expression in Cancer. So I'm gonna talk about my project and about my PhD project, but I'm also gonna share with you like some of the takeaways, some things that haven't been like really great with my project, because I think and this is something that we really have to thank Alice. I think we've grown like a pretty safe place to share things and problems about PhD students. So yeah, the talk is gonna be kind of like that. So I hope you like it. So this is the Computational Systems Biology Lab at the National Institute of Genomic Medicine in Mexico City. This photo was taken like 2 years ago, I think. So it was really crowded at that time. I think that right now we're half of them. A lot of the students have been graduated and everything. So yeah, I wanted to present them. So for our lab, it's always been networks. It's always been network biology. Our PI is a physicist, so he always tried to put the system biology approach into our study. So yeah, they do have papers back from 2012 and before, way before, that they were looking at biological network from different tissues, from different types of samples, like, and also different types of networks, because right now we do correlation networks, but they also had some Boolean networks studies. So what does it mean to be a computational systems biology lab in Mexico? It's kind of hard actually. So it means that you won't have samples of your own to work with. And I know that I mean that is not the case only for Mexico. I know that that might happen in other places too. But being in Mexico, it also means that almost no lab in the country will have enough samples to work with. To actually build the co-expression networks, we rely on having at least 50, 50 to 100 samples per, I don't know, per subtype, per cancer type, per whatever. But yeah, almost no lab in Mexico have them. So what we do is that we use public databases and mostly TCGA. So, okay, this story goes back to 2017 when I was working as a software engineer in a company called Thermo Fisher Scientific. Uh, Thermo Fisher, it's kind of it's the Home Depot for laboratories. They sell like everything from sequencers to pipette to like they sell everything. So yeah, I was working there, developing a project, a part of their cloud tools. It's like the Dropbox. It's a place where you can upload files and everything, and then you can run some experiments. Yeah, they're trying to connect all of their instruments, all of the instruments that they have available. They're trying to connect them to the cloud, so you have only one entry point to actually manage everything you have, everything you could have in a lab. So yeah, I was working there and I was kind of happy, but then I realized that I miss reading about biology and reading papers, and I miss the academic life. So I wrote an email to Jesus Espinal, that he's now my PhD advisor, and I was like, hey, do you have like a nice project that I can work with for my PhD? And he sent me this picture, and he said, uh, "This is healthy." I don't see. I don't know if you can see my pointer. He said, on the left you have healthy, on the right you have breast cancer. Like it's different at plain sight. And he said, do you want to work with this? And I was like, yeah. I thought it was like really interesting, um, because of like because of the difference, only because of the difference. I didn't actually knew what these circles plot were about, but yeah, he showed it to me. I was like, yeah, let's do this. So we proposed a project to um, to investigate this, and it's all based on a paper that they published in 2017. The paper is called loss of long range no, sorry, the paper is called RNA-seq based genome wide analysis reveals loss of inter-chromosomal regulation in breast cancer. Yeah, it's like a really long title. I don't like it, but it is about loss of long range co-expression in breast cancer. So what they did for that paper is that they took TCGA samples, tumor and healthy samples. They did normalization, they did quality control, they did everything. And then they they got differentially expressed genes, and also they run cytoscape to get they run RNA to get the network. Sorry, I think someone has their micro open. I don't know if they have a question or something, but yeah, you can ask me questions at any time. So yeah, they run RNA to get the co-expression networks, and this is what they found. They found that the healthy network, this is again the co-expression network derived from RNA sequencing samples from TCGA, and this network has the top 10% from the most like the highest values of mutual information. Mutual information is the measure that we use to get the co-expression values. So this network has, each node is a gene, and the most co-expressed genes are connected, and genes are colored by their by the chromosome where they are located. So this is the co-expression network for the healthy phenotype, for the healthy samples, and this is the co-expression network for breast cancer. Again, the colors are chromosome, and as you can see, the healthy network, it kind of looks like a confetti. You have genes from different chromosomes interacting with genes from other chromosomes, and in cancer, you have connected components that belong mostly to only one chromosome. So yeah, that is like the main findings in the in the paper. Then they got some signaling, some important signaling pathways, but none of us likes this part of the paper, so we never talk about it. And yeah, so final considerations for this paper, they got a systems biology approach to unveil sort of relationship at the transcriptional level in breast cancer, and the results were validated by independent datasets. So the paper kind of finishes like this. To our knowledge, this is the first time that the RNA sequencing-based network inference in breast cancer reveals loss of inter-chromosomal interaction. And as I mentioned before, the lab had been working with biological networks like for a very long time. And I always ask, like, why was the first time that you saw this, like with this paper? And it's actually because of the RNA sequencing data. So before, I don't know if you know uh, about the cancer genome atlas database. When they first released their data, they were microarrays, and then they compile everything in a new data set that we're gonna see right away. It's called, the database, it's called Genomic Data Commons Data Portal, and they actually resequence everything. They resequence all of the samples, and now they had RNA sequencing data. So yeah, that is why we saw these patterns for the first time. So okay, the next part, we already knew that this was happening in breast cancer, so we wanted to see if this was, if the parents were also present in breast cancer molecular subtypes. So yeah, this is the first paper associated with my PhD. The name is Gene Co-expression is Distance Dependent in Breast Cancer, and it was published on July this year. So, okay, the first paper was published in 2017, and this one was published like just a few months ago. So, why did it take us so long to publish this? It didn't take that long to publish. It did take a lot of time to actually write it. We did have the results, and I don't know if it was because I was like. Um, a fresh new year PhD student, and I'm also the first PhD student for my advisor. So this whole thing has been kind of an experiment. So yeah, we will get some results and we will draft something, and then we will share the outline with our coworkers, and they will say this kind of doesn't make sense. So we will do it like we will do it all of it again. I had like so many figures from like different results and looking at it like from different perspectives. And so yeah, that is why it took us so long time. So finally we got it done, and this is actually the pipeline that I set up for the project, and it's the pipeline that I'm using. So we use, as I said, we use data from the Genomic Data Commons data portal. They have samples from many tissues. They also have DNA sequencing. They have RNA sequencing. They also give you like some data that is already processed. They have clinical data. They have a lot of data. So our pipeline goes like this. I extract the data, then I perform normalization. Then we compute mutual information as I already said, as a measure of co-expression, of gene co-expression. And then we have two different kind of pipelines. In the first one, we use the entire set of interactions, the entire set of co-expression values, and we do distance co-expression analysis. And on the other pipeline, we only get the most significant interactions, the top interactions in the entire set. And we did this because we didn't want to rely on like thresholded networks. Um, I think I mean, they are okay because we see the results and everything, but yeah, no one is like really excited about uh, thresholding thresholding networks. So that is why we also use the entire set of co-expression values. So yeah, with this entire set of co-expression values, we saw that there is a decay on the co-expression values in breast cancer molecular subtype, and that this decay is dependent on physical distance between gene pairs. So we have here the breast cancer molecular subtypes. The four main breast cancers are called luminal A, luminal B, HER2 enriched, and basal subtype. And as you can see, for very close, like physically close genes, their co-expression values are really high, and then there is a decay, and then they reach a plateau in the breast cancer molecular subtypes. But for the healthy phenotype, we don't see this decay, and the values remain more or less regular. So here we have beans, distance bean of five megabase pairs, and the line is for the mean value, and then the highlighted part is for the deviation. So yeah, we have this decay, and then we wanted to know if the decay was different for each breast cancer molecular subtype, and it actually is, but it is also different for each chromosome. Here you can see the healthy phenotype and also the basal subtype. And here we have beans for 100 gene pairs. And as you can see in the healthy phenotype, there is almost no variation in the mutual information values for these genes. Uh, and it is different in the basal subtype, where you actually have this variation between the values in chromosomes. So yeah, for to be able to compare the MI values between breast cancer subtypes, we normalize them, and yes. So that is for the entire set of interactions. But now for the networks, or for the top interactions, for the most co-expressed pairs of genes, we see in this circus plot that there is an intra-chromosomal interactions proportion imbalance. So the orange links connect genes in different chromosomes, and the purple links connect genes in the same chromosome, and the blue ones connect genes in intra-cytoband, uh, in the same cytoband inside a chromosome. So as you can see in the healthy in the healthy phenotype, there are mostly interchromosomal interactions, and that these interactions are lost for the breast cancer molecular subtypes. And in breast cancer, there you have a lot more of intrachromosomal interactions and intracytoband interactions. So we also saw that the intracytoband interactions are important because they kind of they they allow hotspots to emerge in like very localized regions. And we saw that some of these regions are actually associated with already known alter regions in breast cancer. In this case, we present the chromosome 17. This chromosome is really important, especially for the HER2 enriched breast cancer subtype, because it is where the HER2 gene is located, and almost all of the all of the samples that are categorized in this breast cancer subtype, they have highly expressed HER2, and some of them have also copy number variation, amplification peaks for these genes. So these are the most important results for that paper, and at the end, I did like it after 2 years or so of working with it. I actually did like it. It was mostly about structure. We got the result that we were looking. We wanted to confirm that the loss of transregulation was different for breast cancer molecular subtype, so that was okay. And we also observed, as I said before, the association with breast cancer known copy number alteration, but we did not any more data to it. So after that, we wanted to know if there were some functional implications associated to these structural and topological features in the gene co-expression network for breast cancer. So right now, that is what I'm writing about. I'm almost done with with this paper, and this is not gonna be the title, but it is about loss of long range co-expression in the luminal A subtype, but with functional characterization and with data from other omics. So yeah, this paper has again the networks. As you can see, we have the confetti in the healthy network, and in the luminal we see luminal 8. Sorry, we see a big connected component, and then the smaller connected components that are associated to only one chromosome. And what we did to evaluate this loss of interchromosomal observation for this case, we got communities in both networks, and then we took the intra-chromosomal links per community as a measure of chromosomal assortativity. So we counted on each community. We counted how many genes were linked with genes from the same chromosome, and how many genes were linked to genes in different chromosomes. So we got these distributions. As you can see, the luminal A network has almost all of their communities have a fraction of interchromosomal links very close to one, and it's completely different in the healthy phenotype, although it also has some intrachromosomal communities in the network. So after we extracted the communities, we perform an overrepresentation analysis using the biological processes in gene ontology. Here you have the number of communities for the luminal A network. We have 87 interchromosomal communities and 612 intrachromosomal communities. These intrachromosomal communities are the ones that have a intrachromosomal fraction of links equals to 1, so they have chromosomal assortativity equals to 1. And we perform this enrichment analysis on the entire set of communities, and we got that 20 interchromosomal communities were enriched, and only 9 intrachromosomal communities were enriched. So despite having a greater number of intrachromosomal communities, the majority of enriched communities are actually interchromosomal, and they're associated with a lot of biological processes, a lot of different biological processes. A lot of them have to do with cell cycle regulation and regulation of transcription.
So,
So,
Deanna, we have a question or two questions from Nick in the chat. He's asking, can you explain again the physical distance that you plotted? Somebody lost sound. Deanna, can you still hear me?
Yeah.
Okay, so Nick is asking, can you explain again the physical distance that you plotted the mutual information against? He wasn't quite sure what the plots were in the slide where you talked about chromosome 17.
Okay, so the physical distance, it's right here, and it's only the distance in base pairs. Yeah, here or here is the distance in base pairs between the two genes that interact. Yeah, it's only where does the gene start, and where does the other gene start.
Okay, thank you. That makes more sense.
Thank you. Sorry, yeah. sometimes I think that when you talk about your project like for so long and so many times, you ended up like not saying the most important things because you already have them in your head and you forget to explain them. Yeah, sorry.
I had one more question.
Okay.
On the slide before this one. Yeah, this one. Can you explain again? I'm sorry, I was having a little trouble understanding what the diagrams were.
Okay.
Can you explain it again?
Yeah, sure. So we have the networks for the circuit plot. We have the most significant links in the mutual information datasets. So in this case, we have inter-chromosomal interactions and intra-chromosomal interactions. The inter-chromosomal are in orange and the other ones are in blue and in purple. So to get to this figure, we took only the intrachromosomal interactions, the interactions that are inside one chromosome, and this is actually chromosome 17. So you have the diagram of the for the chromosome, and these are like chromosome regions. They are called cytobands. So yeah, what we found is that some of the hotspots, like some of like high density regions for intrachromosomal interactions are actually associated with important regions or regions that have been previously reported as altered in breast cancer. But these are only for intrachromosomal interactions.
Oh, okay. Thank you. That makes so much sense.
Thank you. I'm so sorry.
No worries at all.
Okay, so yeah, so now we have again the network for healthy and for the luminal A, and I was talking about the enrichments. Okay, yeah. So in this plot, we have the chromosomal assortativity. That means how many interactions are from the same chromosome inside a community, and then expression assortativity. And the expression assortativity is that we got a differential expression for the genes in the entire network, and then we added a label if they were upregulated or downregulated, and again we counted the fraction of links that were connected to like same label of differential expression in on each community. So we got some communities that are highly, let's say, same level expressed, and they're also connected with links that are from interchromosomal interactions. As you can see here, the majority of communities that are actually enriched, that are associated with biological processors, they are in the first quadrant of the plot. So this means that their genes have like the same differential expression, but they are located in different chromosomes. We also have one highly overexpressed community that is formed with links from kind of the same chromosome. But yeah, the majority of the enriched communities are actually interchromosomal, and their genes are differentially expressed like in the same the same sign, let's say. So after seeing that, what we wanted to get was if there were if genes if transcription factors were actually participating in these differential expression patterns that we saw. So to evaluate that, we downloaded a database that is called Gene Transcription Regulation Database. And for each community, we identified the transcription factors that were present in the community, and we identified their regulated genes, and the communities that actually have transcription factors associated are these ones. And kind of an important community is the NUSAB1 community. The communities have the names of the genes with the highest phase rank. And the NUSAB community is this one that we have in part B. It has some transcription factors that have previously been reported like as very important for breast cancer, such as this gene called SEMPA and this gene called FOXM1. This gene is actually associated with the luminal fate, or with luminal cell fate. So as you can see in this community, almost all of the genes are upregulated. So it does has a high expression assortativity value. And yeah, and this one is the one that has the most important transcription factors. This one is a community, is the NR4A1 community. It also has it has 4 transcription factors, but it doesn't have a lot of transcriptional regulation interactions. As you can see in the NUSAB1 community, there are a lot of genes that have the circle, the border in yellow, and these genes are regulated genes. So in this other community, actually there are a lot of interactions among the transcription factors. And another important community is the community that has a big set of riboproteins, and this was actually kind of this was new because I really looked for reports about expression in luminal A in particular, but in breast cancer for ribosomal proteins, and yeah, they've seen some ribosomal proteins are underexpressed, but not like a whole cluster. And so these ribosomal proteins are located in different chromosomes, but in our network they are placed in the same community, and they also have some transcription factor, but these transcription factors are not ribosomal proteins. So again, we have, I mean, this is mostly for inter-chromosomal communities. And now for the intra-chromosomal communities, we wanted to see if the hotspots were associated with CTCF binding sites. As some of you may know, these proteins are important for the structure, for actually DNA 3D structure. They form what are called insulators. That these are regions in the chromosome that are used to like get close one promoter to their regulated genes. So yeah, when we show this plot, it I don't remember exactly who that was, but yeah, they told me you should look for CTCF binding proteins at the boundary regions that they may be there, but actually no. Intrachromosomal communities are not bound by CTCF binding sites. What we did to evaluate this is that we took the regions from the communities, and we set up a 50 kilobase window, and we counted how many binding sites were at the boundary regions and how many binding sites were in the middle regions. And to do that, we got an previously published data set for chip-seq analysis. And yeah, when we counted the binding sites in the boundary regions and in the middle regions, I got this. So yeah, you have for some of them, for some communities, the total number of CTCF binding sites are more actually at the end region of the community. But yeah, out of the 416 intrachromosomal communities with CTCF binding sites associated, we got 197 communities with more binding sites at the boundary regions and 219 with more binding sites in the middle regions. So we also check amplification and deletion peaks using data also from Genomic data commons and running the GSpeak algorithm. And we perform enrichment analysis with the intrachromosomal communities to see if they were associated to these altered regions that we also saw in the previous papers. So we actually got that interchromosomal communities are enriched with deletion peaks and not where with amplification peaks. And this was actually kind of surprising because we thought we were gonna have more amplification peaks because that might be something that could like when you have the gene, well, you have the chromosome, and then you have a region that is amplified, and this region, I mean, this region has more than two copies in the chromosome. So if the region actually expands more than one gene, what you can have is like more co-expression values in those regions that are close together. So that will be something that will explain the co-expression values that we got. But yeah, this is not what we're seeing. We're actually seeing that deletion peaks are more associated with intrachromosomal communities. So this paper is kind of I know that some papers they said that they're like we find, but this one is actually kind of we did, but we didn't find. So yeah, there is a contribution of transcription factors, but not not every single intrachromosomal community or interchromosomal community in our network has transcription factor associated. So yes, there is also communities with differential expression, but they are not, I mean, some of them are overexpressed like in a in the same fashion, and some of them are underexpressed, but they don't. I mean, it's not something that is shared for all of the communities. And no, the CTCF do not play a role in the interchromosomal interactions, and the hotspots are related to deletion peaks. And actually, yeah, the biological processes are associated in this phenomenon, but only for interchromosomal communities. So yeah, this is for the luminal A breast cancer. And now for other types of tissues, this is the loss of long range co-expression in other tissues. These are the results, but yeah, I haven't started writing this preprint. I should do that. And yeah, so what we wanted to see was if this phenomenon also holds in other types of tissues. So so yeah, first we check if the decay in mutual information value is also present in other tissues, and yes, it is present. Each manifestation displays its particularities, but they all have a decay, and this decay ranges from like very abrupt in lung cancer to almost imperceptible in thyroid cancer. So the phenomenon is qualitatively shared, but the quantitative elements associated are different. And we also wanted to see if the networks display the same topological features. Yes, they do. Again, for this network, we took only the most significant interactions. So there is a constant display of opposite characteristics in the co-expression networks in all tissues. And what we're actually seeing is that the healthy networks, they do share fundamental topological features. They all have one big cluster, one big connected component, and then smaller components composed mostly by inter-chromosomal interaction. So that might be associated with a robust and stable behavior in the transcriptional program for healthy cells. So we also wanted to check if there are hotspots in interchromosomal communities in these different types of tissues. And yes, they are. They are also different, different for each tissue. And as you may have noticed, the kidney cancer has uh, significantly more interactions than the other types of tissues, and this is something that has to do with the sample size. So to get these networks, as I said before, we get the most significant interactions, but that p-value associated to the to the gene interactions, uh, the p-value has to do with the sample size. So yeah, we observe intrachromosomal hotspots, and we also check for community structure in these intrachromosomal interaction networks. We look at the distribution of the mean distances in the communities, and we see that this distribution tends to go like to really small distances. And we also see that in the healthy network, but not as severe as in cancer. And this happens for all of the cancer types. So short distance is preferred also in the healthy network. So finally, these observations suggest that there is a loss of long range co-expression, and that this is a common feature in cancer. So do we know why is this happening? No, we actually don't know. And do we know if this is a cause or a consequence of the process of carcinogenesis? No, we actually don't know. And for me, it was something uh, like it's something that actually kind of stressed me, because I was like, how am I gonna talk about a phenomenon that I don't know why it's happening? But then this semester, I didn't mention, but my background is actually in computer science. So this semester I'm finally taking a molecular biology class, like a real molecular biology class. And my professor said something that kind of like it stuck in my head. He said that for biology, it's a lot at first observation, then description, and then measurements, and at the end you kind of get an intuition of why is a phenomenon happening, and that this is something that has been with biology like for forever. We were looking at the at the at the experiments that got to the DNA structure model. So yeah, so now I think that for the loss of long range co-expression phenomenon, we're in the description and measurement part. So I hope that the intuition comes later. So until now, we know that this phenomenon is consistent, is measurable, and it has a reproducible effect that is present in all the co-expression profiles it studied. This is there is a global alteration of the interactions in the transcriptome where we are looking at how the transcriptome is interacting. So long paths are lost and shorter parts are preferred for the cancer transcriptome and also for the health transcriptome. We see that there are long-term implications in the phenotype, or at least we know that these phenotypes are different. We know that we're seeing cancer and that we're seeing healthy. So we kind of hope that these are that these difference that we're seeing in the co-expression patterns are kind of implicated to the phenotype. There is a contribution of the multiple alterations in the regulatory mechanisms of transcription. As I mentioned, it's not only copy number alteration. We also think that there may be some alterations with the 3D structure in the genome, and also that may be some epigenetic marks associated with this phenomenon. And to actually evaluate that, we are currently added other omics data, and the prevalence of the phenomenon suggests an enabling feature in the transcriptional program. So yeah, we're still in the path to evaluate this phenomenon that we name loss of long range co-expression. So that's actually all. Thank you very much for your attention.
Yeah.
Thank you, Alice. Sorry, I can't see the chat.
So
Yeah,
I think that there were some comments like on what you said, that the next questions you had addressed already.
Okay.
So I'm going to start off in this Q&A. So first of all, this was really great. I did start out in kind of computational biology, network science at the beginning of my PhD. It took me kind of back to the mindset that I was in a few years ago. And so one of the things, like starting on a more technical end, because I think you were very much focused on a lot of the technical stuff, so that might be things that you're interested in, is I saw that you used mutual information to construct the network. And perhaps that's a thing that came about recently because I haven't caught up with the literature in the last few years. But like when I was still working on it, it seemed okay to use things like correlation. And I know that mutual information is a lot more difficult to compute, and there's like some parameters that you need to set. And so I was wondering what kind of motivated you to do that. Like, is there some advantage that you get from just compared to using simple correlation?
So we've seen the correlation patterns using Pearson correlation, and actually the phenomenon is the same. We use mutual information, I think, mostly as a, I don't know, our PA started using it like many years ago. So it was like, okay, if you want to compute co-expression values between genes, you use RNA is the algorithm that we use. So I think it's kind of more of like a tradition than something else. But yeah, I do want to evaluate other correlation measures to see if the phenomenon holds. But yeah, if you use Pearson, it's the same.
Okay, I'm relieved to hear that. So we have I think a raised hand from Mason. Do you want to ask a question?
Yeah, thank you. I'm not very familiar with the type of data, so I'm wondering how much temporal resolution you have in this RNA.
Okay, temporal resolution. So the RNA samples, I mean, they go and they sample a tumor in a very specific, I mean, in a day, and then they get the RNA sequencing data. So what we're seeing here is like a picture for many tumors, and as we know, cancer is like really heterogeneous, and these samples could have been like taken in different, like um, in different times in the lifetime of the tumor. So yeah, actually, time is something that we cannot talk about because we only have like a big picture of the transcription characteristics of a sample. But what we can actually do, and it's something that a friend of mine in the lab is doing, he's taking as just like I took breast cancer subtypes, he's taking stages in cancer, and he did that for, I think, for kidney cancer, and the results, yeah, that paper is going to be published soon. But yeah, they don't see the phenomenon as clear as you can see it when you compare cancer between healthy, and if you compare like the different stages in cancer, you don't see the phenomenon that clearly. But yeah, time is not something that we can actually associate.
I have a follow-up question then.
Okay.
So now let's do a follow-up. So when you're talking about primarily the transcription of the genes, but can you tell anything, or there's data on, is that because of the gene, the DNA themselves that's changed, or it was just a change in co-expression?
So I think that the main alteration that we see are actually in co-expression, but when you measure RNA-seq, I mean, when you measure the transcription, like you kind of have all of the alterations, like if you have many copies of a gene, of if you have a gene that has been deleted, of if you have something that has changed in the three-dimensional structure of DNA, you're gonna see all of that alterations actually reflected on transcription. So yeah, what we see is differences in transcription, but there could be like a lot of differences in the mechanisms that actually regulate transcription that we're not actually seeing because we can only take like the last part of the cells pipeline.
Thanks.
Thank you.
We have another question from Nick.
Yeah, I have a bit more of a basic question.
Okay.
So this is not at all my area, if you couldn't tell. But I thought it was really cool. So thank you for a great talk.
Thank you. Thank you.
I learned a lot, and I was kind of curious. So when you presented those networks, like how can you explain again how you quantify uncertainty? Because I'm assuming if you have all this data, it's not gonna like match exactly. And as you said, there's different stages of cancer too.
Yeah. So yeah, uncertainty. Okay. So as I said before, like the RNA sequencing and actually the networks are kind of like a picture in one moment, like from a lot of things that are going on inside the cell. So yeah, uncertainty, I don't think that it's something that we can actually measure and translate it to the network. What I mean, the only thing that we can do is associate the significant, a value of significant, a p-value that tells you if the co-expression value is significant for your samples or not. And they actually, if you see like co-expression values that are really high, they're mostly gonna be significant. If you see co-expression values that are like pretty close to zero, those are the ones that are left behind from our network. So yeah, I think that's the only thing we can do about uncertainty, because yeah, there's like a lot going on in the biological and also in the technical part of this.
Okay, I think that clears it up for me. Thank you.
Thank you.
So I have one more question, Liana. So I was really interested, to be honest, when you started when I read your title, because I didn't know what the space in a gene network was going to be. And so I saw that your space is in the mega base pair. So how far away are two genes if you kind of just uncurl everything, put it in a line, where are the
Yeah.
things? You know? So, but that only works within one chromosome, or does it work kind of with the entirety of it?
No. That only works in one chromosome. Yeah, distance is only it's only you can only measure it for one chromosome. And actually the first figure that I showed you, uh, yeah, the one that has the decays. Yeah, this one is actually looking only in intra-chromosomal interactions, because yeah, you cannot measure distance between genes that are placed in different chromosomes.
Can you, so you mentioned in the beginning, at the end, that something like physical distance might be interesting too, right? Because the DNA is kind of curled up, so there might be things that are physically close, but they're not close in the kind of uncurled megabase pair distance notion. Is there like some data that you could use to evaluate whether physical connections, long range or short range, are affected by cancer development?
Yeah, actually there are some experiments that are called high-C,
Mm-hmm.
where they actually measure how close are some regions in the DNA. Yeah, how close they are. And they get these, or also these matrices, uh, where they say if they are like really close, it's a highest value, and everything. But we actually, yeah, another student at the lab is looking at that type of data, because we actually want to see if the high co-expression in value interactions that we see in genes from different chromosomes, if they actually if they are like physically close in the
Mm-hmm.
genome. I mean, we know that if genes we know that genes that have like really low distance in base pairs are close. I mean, if they are in the same chromosome, like they are close. But for genes in different chromosomes, we actually don't know that. And yeah, there are some data, but I don't know if the problem with this is that we do have, and I mean the genomic data common has, like, as I said, a lot of data, but they do have it for cancer samples. But there's not a lot of labs and a lot of people looking at healthy samples. So yeah, when you want to compare things like this, it's difficult to get healthy samples to actually see if what you're seeing in cancer is like really really
Hmm.
really significant.
Thanks, Diana. That was quite enlightening. Thanks for your talk. There were lots of comments about how people have learned from it. We're well over time already, so I'm going to end this here. But thank you again, and for those of you still on the call, I hope to see you again next week.
Thank you so much.
Bye.
Bye.